Yes, I welcome all of you to our today's talk on BTL 5 to 7 Introduction to Ecological Chemistry. And meanwhile, you received lots of lectures. Today, I shall discuss on another topic and also give you a review article published on bioactive natural products. And from that review article, you need to find out as at least three uh, phenolic compounds, three alkaloid compounds, three terpenoidal compounds with their biological activities. Their source, name of the compound, their source, their biological activities, and the structure. So what you can do, you can cut and paste some of the information and make a table. So first you can put three phenolic compounds, then three terpenoidal compounds, and then three alkaloidal compounds. So then you put other information according to the compound and go through that very fantastic review. So let me show you that review whether it is available on my desk or not. First, show me. Yes, this review. Can you see the slide?
Yes, sir.
Okay, very good. You can see it is a chapter of a book titled Biochemistry and Molecular Biology of Plants, a very important book published by American Society of Plant Biologists. And this review focused natural products, especially secondary metabolites. And you can see it described different kinds of secondary metabolites from terpenoids, alkaloids, phenylpropanoids at their biosynthetic pathway, and all other things, flavonoids, even metabolic engineering. So please go through this review and you can find interesting information that I asked you to extract from the this review. So it is a nicely written and a comprehensive review and some of the historic information about the use of alkaloids by the ancient people, like conine alkaloids have been used to kill the prisoners in ancient Greek, which is biosynthesized in conium maculatum. Here you can see the illustration of the great philosopher Socrates, who was poisoned by hemlock. Hemlock, actually, the chemical composition is an alkaloid called conine, and it was injected. And gradually, how nervous system was collapsed, it is described and illustrated by an artist. So lots of other information you can find, different ancient product, anti-poisoning, nostrum containing opium, wine, snake meat is still used today in rare instances, that is uh, teriac. So natural products have been used by many ways, and still natural product is a big treasure box for the industry, for the pharmaceutical industry, and saving our life in many ways. So please go through this review and you can find other things engineering natural product, because natural products in most cases they are small quantity in the cell of the producer, but for industrial purpose you need larger quantity. In that case, after the discovery like pheromone, uh, organic chemist synthesize the compound, interesting compound in the laboratory, and then industry produce it in a large scale for commercial use. Some of the compounds are very difficult to, synthesize in the laboratory. In that case, genetic engineering of the organism uh, is one of the way. For example, a plant bioactive natural product from plant sources. If plant is a rare plant or slow growing plant, in that case, you can use tissue culture to produce mass profuse amount of tissue and extract the compound, or you can introduce the gene into an organism, even a microorganism, to produce large quantity of the compound like insulin. Insulin has been produced industrially by using yeast and bacteria. So it is a fascinating article. Please go through it. And today I shall share with you this article. But today I want to coin a very interesting topic, phytoalexin. Phytoalexin, I am sure you will be excited to know that class of compound. Uh, Phytoalexin, yes. Phytoalexins, all the plants, microorganisms, and many other organisms, even vertebrates, they produce different type of defense molecules or chemical substances for communication, for interactions with other organisms, even the members in the same group, for their fitness, communication, as well as their, dominance in the ecosystem. Most of the compounds are constituent. That means constituently they are present in the cell. Like a country has, some arms and ammunition for its protection. But there are some compounds which are not, in normal condition, they are not present in the, for example, in the plants. But when they are attacked by uh, herbivore, or they are under a severe stressful situation, to tackle the stressful environment, they biosynthesize very quickly, rapid biosynthesize new secondary metabolites, or some biochemicals. They are called phytoalexins. So phytoalexins are considered uh, one of the plant immune system. For example, we are now ready to take coronavirus vaccine. What vaccine will do? Vaccine will enhance, trigger the cell to synthesize antibody against the SARS coronavirus 2. Similarly, uh, you can, by using some stressor or some trick, even phytoalexin synthesis can be enhanced. So phytoalexin is associated with plant immune system, and understanding of phytoalexins would help us to do some vaccination in plant or like that. uh, One of the common understanding you have now, that plant experience various kinds of stresses as human do. So stress is common. There are lots of several causes of plant stress, and stress is an elicitor. because when stress can be sensed by the plant, this is why it is called elicitor. A plant can sense and try to defend uh, uh, against the stress. Uh, One of the important group of elicitors are insects. Insects, are a large diversity. Insects have a large diversity, and they are depend on the herbs, that is plants. Whether another stressor, drought, flood, wind, hail, even salinity, like that heat stress. Ethylene, one gas, it is also a stress. Ethylene gas can cause stress at parts per billion level. Post-harvest storage, when you harvest a plant and you store them, they are in the stressful situation, not in the natural condition. UV light, another stressor. UV light is is an abiotic stress for all organisms coming through the sunlight. Physical wounds, harvest, transport, microbial infection, plant pathogen can, when infect the plant, they also cause serious stress to the plant, and they are also elicitor. So all these stressors or elicitors, when they are in contact with plant, plant also can sense and can respond in different way. One of the way is to produce small secondary metabolites, sometimes big molecules, to defend the stress. So classification of disease resistance factors in higher plant. In higher plant, there are disease resistance, especially disease is caused by different kinds of microorganisms mostly, but disease is any abnormal physiological situation of an organism like higher plants. So it can be caused by non-pathogenic non-living organisms, also due to environmental reason. Uh, Pre-infectional compounds, there are some compounds which are involved in pre-infectional responses. They are called prohibitins, metabolites which reduce or completely halt the in vivo development of microorganisms. For example, wheat blast fungus, some of the uh, genotype we have, for example, GR119, Albanian genotype. Uh, it is 100% immune, so it might contain some of the metabolites or some other mechanism. We don't know exactly the mechanism. Uh, Obviously, there are some metabolites that completely halt the in vivo development of my blast fungus when you inoculate, even in an ambient condition, but we didn't see any symptom, or very symptom is very limited. Inhibitins, metabolites which undergo post-infectional increase in order to express full toxicity. Metabolites are present there in small quantity, but when plants are undergo infection, infection by the microorganism, that immediately they increase the dose, concentration, or amount of the particular metabolites for expressing full toxicity. Because dose makes the poison. Dose is very important for showing the full toxicity and stop the or inhibit the microorganism. Post-infectional compounds, there are also two types. Post-inhibitins, metabolites formed by hydrolysis or oxidation of pre-existing non-toxic substances. Actually, it is very quick, just hydrolysis and oxidation of pre-existing compounds, and that compound are used for post-infection inhibitory activities. And the last one is phytoalexins, metabolites formed de novo after invasion by gene expression or activation of a latent enzyme system. What does it mean, de novo? De novo means when plants are infected by microorganisms, it can sense and immediately a particular gene is expressed, and due to the expression of the gene, a particular inhibitory substance is produced, or some latent enzyme system. That enzyme system was inactive, but sensing the uh, pathogen toward of it, latent enzyme system becomes activated to produce a new molecule. That means that molecule was not present in normal condition. Uh, when plants are affected by any microorganism or insect or any other stress, sensing that stress, specific particular gene associated with responding to that stress are expressed and metabolites are produced. They are called phytoalexins. So phytoalexins are very interesting, and understanding about the um, phytoalexin biosynthetic pathway and which phytoalexins are producing by which plant are fundamental for developing the biorational management system against the pathogen or any stress. So phytoalexin, phyto means plant and alexin means to ward off. So this word comes from two words actually. To ward off means to stop or to limit the uh, particular stress or particular stressor. They are low molecular weight organic metabolites produced by plants in response to stress, and they are self-made antibiotics. Very interesting. But human has no such ability. What we can and do in our case, antibody is produced, not like low molecular weight organic metabolites like phytoalexin. And more than now more than 300 true phytoalexins are known. The phytoalexin concept is not new. It has been first proposed by Mueller and Boerger in 1941. They gave a theory. Their theory was a phytoalexin is a compound which inhibits the development of the fungus in hypersensitive tissue and is formed or activated only when the host plants come in contact with the parasite. The defense reaction occurs only in living cells. The third theory, the inhibitory agent is a discrete chemical substance, a product of the host cell. Fourth, the phytoalexin is non-specific in its toxicity towards the fungi. However, fungal species may be differentially sensitive to it. The basic response in both resistant and susceptible cells is the same. This is very interesting. The basic response in both resistant and susceptible cells is the same. The basis of differentiation between resistant and susceptible hosts being the speed of formation of the phytoalexin. What does it mean? In case of susceptible one, phytoalexin synthesis is slow and very weak, but obviously phytoalexin is produced even in the susceptible, and this is why the pathogen can override. But in case of resistant, the phytoalexin formation system is very rapid and quantitatively high to limit the pathogen. The defense reaction is confined to the tissue colonized by the fungus and its immediate neighborhood, not the whole plant. The resistant state is not inherited. This is interesting. It is developed after the fungus has attempted infection. The sensitivity of the host cell, which determines the speed of the host reaction, is specific and genotypically determined. So, uh, if we go back, there are seven components of the Mueller and Boerger theory. They actually studied uh, with potato fungus, and at that time, in 1941, the phytoalexin was not discovered by them, but they gave the theory based on their observation without purifying a genuine phytoalexin in the infected tissues of the potato. But later, this theory was fully established. So, production of phytoalexin may be stimulated by certain compounds called elicitors, high molecular weight substances found in the cell wall, such as glucans, glycoprotein, and other polysaccharides. For example, in case of fungus, fungal cell wall consists of, glucan, chitin, or other polysaccharides. So these polysaccharides, when fungus get in contact with the plant cell can sense and recognize those cell wall, that means can sense the presence of an enemy, and they are called elicitors. So elicitors are molecules, high molecular weight molecules that elicits or induce the synthesis of phytoalexin. Uh, it can be a gas also, like ethylene. Uh, Production of phytoalexin in susceptible plants, a pathogen may prevent the formation of phytoalexins by the action of suppressors. This is another term produced by the pathogen. So phytoalexin naturally, in susceptible and resistant plant, both can sense and respond to synthesis of phytoalexin. But in case of susceptible plant, pathogen may use a counter, chemical uh, measure to suppress the synthesis of phytoalexin. Those molecules are also suppressors, are also big molecules like enzyme or glucan, glycoprotein, or a toxin produced by the pathogen. So phytoallergic toxins in our food, there are huge. For example, in case of potato, cinnamate, scopoline, quinic acid, sesquiterpenoids, solanine, very famous, solanine and chaconine, they are present in our food, and we are getting them through the food. Solanine is a glycoalkaloid and very, very toxic compounds. Sunburned spots or growth shoots, when potato, is a modified stem. If it is sunburn, you can see it turns into uh, uh, a greenish color, and it allows to synthesize huge amount of solanine, triggers to synthesize huge amount of solanine. Solanine 10 to 50 ppm is normal and increases sevenfold during stress. And natural pesticide, cholinesterase inhibitor, it is a cholinesterase inhibitor. Acetylcholine is a neurotransmitter, and extremely bitter. That part of potato, if you test, you can find bitter and not soluble in water, and it is heat stable. So when you buy potato, never buy the potato which is partially green or like that, due to exposure to the sun, because glycoalkaloid solanine is highly toxic to human also, and it can inhibit the uh, neuron system, and it is a neurostimulating compound. This is why it is a neurotoxic, we can call, and this is why it is better to avoid. And it is present actually in the tomato, potato, and other solanaceous plant in small quantity, but under stressful situation like sunburn, the quantity is highly increased. Stress metabolites in tomato, cinnamaids, lecithin, phelcarindiol, tomatine. Tomatine is very, very important, and it is an alkaloid high in immature fruit. So, immature tomato you can find beta test, this is due to the presence of tomatine. Ripe tomato contains 30 to 40 ppm of tomatine, and tomatine has some a good effect also at lower concentration, natural pesticide, and it is heat labile. Carrot also cinnamaids, phelcarindiol, and isocumarine. Isocumarine, actually neutral phenolic compound, antimicrobial. Even 10 ppm is active against the microbes, extremely bitter, not soluble in water, heat stable, and ethylene sensitive synthesis. So, what you can imagine that through vegetables and many plant materials, we are always getting lots of secondary metabolites, lots of toxic substances. However, we are not affected by, mostly not affected by those toxins, because they are present in lower concentration, and for the human body, uh, over the period of time they are adapted to a certain level of their toxicity. But it is easily understood that natural toxin we are always getting, and some of them are helping us to fight against the microorganisms and other diseases. Pepper plant produce capsidiol, sweet potato ipo, mia maroon, phytoalexin of celery, parsnip, and parsley, soralins, and grapes, resveratrol, very famous one. It is a stilbene, and phenolic compound, resveratrol present in grapes and even red wine, and it is very good for the health, especially powerful antioxidant, uh, and some other stilbenes also. Alfalfa medicarpine, soybean glyceolin, peas pisatin, and bean pods phaseolin. So all these compounds are antimicrobial, and they are known phytoalexin. Uh, Plant versus man, genobiotic metabolism. Cytochrome P450 system is present in body, liver, lung, skin, nasal mucosa, GI tract, and it enzyme system to solubilize chemicals. And especially when we take genobiotic, genobiotic is a synthetic compound, not naturally available, like DDT. So, in case of genobiotic compounds, we are getting lots of antibiotics are synthetic, and they are, uh, in our body, they are handled by the cytochrome P4 system, different enzymes. They, solubilize these chemicals and through urine or other excretory system, throughout from the body, and sometimes detoxify by using different enzymatic mechanism, and if not possible, then deposit in the liver and encoded, coated by the lipid layer, so that it cannot harm the body. Plants did not want to become food for people or animals, but we fought back. This is true, because plants are only the organism which can utilize solar energy and convert it into the chemical energy. So, for the energy source, all living organisms are dependent on plants, but plant never likes to be eaten by others. So, antioxidant enzyme system, there are lots of enzyme systems are present in the living organism, like you can see uh, superoxide dismutase, glutathione, peroxidase, catalase, because when any organisms, even plant or human, they produce lots of superoxide, and these enzyme system are then work and neutralize the superoxide, because superoxide are highly reactive on the membrane as well as DNA, RNA, and can cause serious consequences even in cancer. So cytochrome P450 system has two phases. One is phase one, what can do oxidation, reduction, and hydrolysis of the compound and make them polar and eliminate from the body, or make conjugation and then synthesis and make it polar. So in case of phase one, oxidation and reduction of the, for example, you have taken an antibiotic, how to release the antibiotic from the body. If it is easy to handle P450 system through oxidation or reduction or hydrolysis, depending on the structure of the compound, phase one is first work, and if it is not possible to make it polar and excrete through the urine or other sweating or other way, then phase two conjugation synthesis system is worked. This is why when you are taking any drug, it is suggested to take much water, so that water is very important. A body has the mechanism to exclude the xenobiotics, which is called the cytochrome P450 system, and this system is present in eukaryotic organisms. Current trend increased consumer interest in nutraceuticals, that is different uh, bioactive compounds from different organisms, including the plants, and lots of nutraceuticals and phytochemicals now you can find in the food as dietary compound or supplement or other drinks, and they are now becoming very popular. And even probiotics are also now becoming very popular, and food supplement is uh, uh, now helping people to fight against different stressful situation, um, and getting slow down the aging process and others. Thank you very much. I should stop here today. So if you have any question, you can ask the question. And we may set a time for your midterm too. When would be the best? Next week? Because you received lots of lectures, so many lectures. So please inform me when would be the best time for you to take the midterm two, uh, preferably early next week. Any question about phytoalexin?
Sir, I didn't understand the part how cytochrome P40 system works.
Cytochrome P450 system present in the eukaryotic cell to how to say to tackle the xenobiotics, especially when you take an antibiotic or exposed to a xenobiotic compound, a human-made synthetic compound, which is are not easily metabolized by your body. In that case, cytochrome P450 system works on that compound because it needs to be eliminated from the body. First, what it does, phase one used to oxidation of the compound or reduction of the compound to make it water soluble so that it can excrete through the urine or sweating or like that. And if it does not work, phase one, then phase two. Phase two means to change the chemical structure of the compound through conjugation to conjugate some radical or like that, or by using some enzyme, uh, you know, uh, it, uh, the structure of the compound is changed so that it becomes polar and it is released from the body. Is it clear now?
Yes, sir.
Okay, thank you. Any more question? Phytoalexin, we discussed about phytoalexin. How can we utilize the understanding of phytoalexin for biorational management of uh, plant biotic stresses in the practical field? Do you have any comment on it? Can you answer this question?
We can also extract from the and also we can also synthesis, then we can apply.
This is one way. The best way would be, if you know the elicitor of the phytoalexin. For example, if you know wheat blast when landed on the wheat plant, then wheat plant immediately produce different phytoalexins. In that case, what you can use non-living, wheat blast fungus, that is the cell wall, what you heat killed or some other way you can kill the fungus and then make it as a vaccine. So you can spray that vaccine as elicitor on the plant and induce the production of phytoalexin. So that would be the smart way. So plant vaccine, uh, commercially there are not such molecules, but there are some molecules. For example, chitosan. Chitosan is a biopolymer. People prepare it from the chitin. So chitosan can work like a vaccine on the plant because plant can sense the presence of a fungus or like that. Although chitosan is a non-living material available in our laboratory. So this type of elicitor type compound, if you can use and trigger the phytoalexin synthesis, especially when you think that plant may be attacked epidemiological condition, if you analyze and if you think that plant may be attacked by the certain fungus, and you can use a specific vaccine against that fungus. For example, in case of wheat blast, it is a head disease. So Magnaporthe oryzae, triticum petotype we have in our lab. What you can do, you culture the fungus in liquid broth, high amount, and then by blending, making them small pieces, and by using formalin or some other chemicals, you can kill the cell, or even you can use the autoclaving to kill the cell, but still their cell wall is chitin and glucan polymer. If you spray on the wheat plant after flowering and induce the systemic resistance, that is phytoalexin and other defense system, that would be a fantastic solution. So you can do even this experiment in the laboratory. For example, some heat killed uh, uh, the MOT fungus you can spray on the leaf. And after 24 hours or 48 hours, you can spray the blast conidia to see whether they are plants are activated to defend the pathogen or not. That would be a very nice. And if you can find such a solution, it would be a really a very big story in science, but it is not yet been tested in case of wheat blast. Any more question?
Sir, is it already available in Bangladesh or it is already introduced in Bangladesh? This
Which
is a
one?
wonderful.
Which one?
So this is phytoalexin or this practice kind of things.
Phytoalexin, Chitosan is available in our laboratory, but phytoalexin present in the plant when they are in under stressful situation infected by the microorganism or insect. So it is not available in the market, but obviously some of the phytoalexins are biosynthesized by the industry, so you can get from the Sigma and others, but they are a bit expensive. And rather than using those molecules, if you can induce plant to make their own antibiotic by using elicitors, that would be fantastic idea.
Okay, sir.
Any
I
Sir,
got
question?
it.
Yes.
sir, we use some nanoparticles in our field, like titanium oxide or selenium oxide. Can they produce phytoalexin production
We
plant
are not
using?
sure, but what titanium dioxide P25 type do, it is a photoactive, nanoparticle. That means in presence of light, it can react with oxygen and make superoxide. For example, hydrogen peroxide, singlet oxygen, or O3, and they are toxic for the fungal cell wall, can kill the fungus. So at certain dose, they are tolerated by the plant because plant has a strong cellulose bound cell wall and but titanium dioxide they produce superoxides to stop the fungus. Whether they are also inducing the phytoalexin in the wheat plant, we are not sure, and it is also interesting question because the plant may sense titanium dioxide as an elicitor and can may produce phytoalexin in addition to the superoxide produced by the titanium dioxide. We are not sure. Phytoalexin, for example, when we use the biocontrol bacteria, plant also induced to produce phytoalexin like substances to activate their immune system. And phytoalexin synthesis is associated with the gene expression. So somehow, if you can activate the defense gene for higher expression, obviously phytoalexin will be synthesized. Any more question?
Sir, can we use phytoalexins for to control pest like pesticide or fungicide in a broad uh, amount? Like that?
Yes, it can be, but phytoalexins, they are antimicrobial substances. So against the some of the phytoalexins discovered by ecological chemists are now commercially being used as drug or antimicrobial substances to protect the plant. Those who you can easily synthesize in large quantity with low cost, and steps of synthesis are very limited. It is possible. And it is like you can use it as a pesticide, obviously. Any more question?
Especially biotic stresses are involved in case of phytoalexin. It's also true for abiotic stresses.
Yes, a good question. Yes, some abiotic stresses also, elicit the synthesis of phytoalexin because some of the phytoalexins also help plants to tolerate abiotic stresses, because when plants are under abiotic stresses, lots of superoxides, reactive oxygen species, are generated in the plant cell. plant becomes sensitized. And those superoxides actually are toxic for the microorganisms. But, in case of abiotic stress, they are not microorganisms. In that case, those superoxides are toxic for the plant own cell, can damage the membrane, or can, affect or hydrolyze or, break down the DNA, RNA, or like that. In that case, some of the phytoalexins, which are, antioxidants, has, hydroxyl group or phenolic substances, they can neutralize or scavenge the superoxides and then allow plant to tolerate, abiotic stresses. So increase the abiotic stress tolerance. Shafi, it's a really interesting question. And the study on the role of phytoalexin in abiotic stresses are poorly being studied. It is an important topic, and we need more knowledge on this subject. But there are lots of examples already been available in the literature. If you browse, you can find it. Now, for example, if you expose plants to certain dose of UV light, then some new compounds which we call the phytoalexin are synthesized, and people have lots of experiences. Even chemical treatment, some chemical, for example, copper oxychloride or some other heavy metals exposure, or even heat stress and other stresses, phytoalexin synthesis is enhanced. So any more question? Very interesting question. If no question, then thank you so much. And tomorrow I shall attend National Core Committee of Crop Biotechnology, because Cotton Development Board, they are trying to introduce BT cotton in Bangladesh. So it is very important expert meeting uh, where my opinion would be important. I shall go there. So your class, scheduled class tomorrow will not be held. What you do, I shall share with you the review as well as this lecture. Please go through well. And the next week, you shall have the midterm true examination. So please suggest me the suitable, date, so that I can prepare the questions for you. Thank you so much.